What's up, Guru Nation? Let's demystify clinical research. Guru Nation, thank you so much for joining. I've been going back to the fundamentals. This one is about randomization visits, also known as baseline visits. The last one I did was about screening visits. You can check it right here or underneath the video if you want. I'll have links to both so you can click. So we're going to go in order. So screening is first. We did 30 minutes on screening. Randomization is next, usually. Randomization goes by baseline or randomization. Before we get to the content, though, look, I do not promote things on this channel a lot unless I use them and I believe in the product. This is Life Boost Coffee, probiotic coffee. It has bifidobacteria in it. I love cold brew. Their probiotics brew is the one I drink, but they have tons of different ones. This one has bifidobacteria in it, which is why I drink it every day. I rarely drink anything else. The link to this video is underneath. Bifidobacteria is the good kind of bacteria, one of the good kinds of bacteria you want in your gut. I learned this from Dr. Hazen years ago. This is not Dr. Hazen's company. It just so happens on another channel I interviewed the founder of this company because of Dr. Joe. We have another channel, the Dr. Joe Show. He recommended me this brand. Not only is it good for you, it tastes amazing. That's why I'm promoting it. I get very little out of the affiliate link. Click on it. Enjoy for yourself. With that being said, okay, let's get into baseline visits. So baseline, synonymous with randomization. Now, not all studies have randomization. Some are just enrolled in open label. All right? Some studies compare two drugs that are on the market versus one that's not, which is the investigational product. Other trials test one versus one, so one on the market, one investigational product. Some have a combination of placebo-controlled in it, so whether it's one on the market versus one investigational product, or two on the market versus one investigational product, or you can throw a placebo in there as well. This is getting into study design, which we're not going to get into in this video. That's an hour-long podcast on its own. But just know that at randomization, you are selecting randomly, all right? That's where randomization comes from. Randomly, the system, which is called an IRT system, Investigational Response Technology System, I believe, but don't quote me on that. It's IRT. Everyone refers to it as IRT. And that's usually where not only do you screen the patient, so this patient's officially enrolled in the study portal, but you randomize the patient in the study. And oftentimes when you hear them, the sponsors or CROs referring to patients being enrolled in the study, they're oftentimes referring to them actually being randomized. So they don't necessarily count screening. They count randomized as actually being enrolled. And most studies are designed to where it's double blind. So what does that mean? It means the coordinator logs into the IRT system. There's a dozen of them, if not more. They already screened the patient previously. So now they're in the system, the same patient. The patient's already been assigned a patient number and everything. The site clicks on that patient. The system will ask, are all the inclusion-exclusion criteria met? Does the patient qualify for the study? If the answer is yes, which we're going to get into what that yes actually means in this entire video, you click a button and say yes. The IRT system will randomly assign that patient to an IP number. So this is when it's double blind or whether it's open label. Obviously, when it's open label, you know what the patient's getting because you can clearly see they're either getting this or that. When it's double blinded, with or without placebo, the staff cannot tell what they're getting and the patient cannot tell what they're getting. So that's called double blind. There are variations of this where we're going to do shorter videos on having unblinded staff, where there might be a few staff members that do know what the patient's receiving, just because it's not practical to hide the identity of the investigational product for a variety of reasons. Normally it's the color of the infusion, but for the most part, they're double blind with no unblinded staff, so that you literally don't know what you're giving the patient and they don't know what they're getting. You know the probabilities of what they'll receive, but you don't know for sure. And that's the whole purpose of this IRT systems, is to give you the kit number so that then the coordinator goes into the investigational product room, the double locked cabinet, grabs the right kit number associated with what the IRT told them to grab for that patient, gets it, and then distributes it to the patient, whether it's a pill, whether it's an injection, whether it's a spray, whether it's an ointment. Doesn't matter what it is. Infusion, it does not matter. The process is going to be the same. So, sounds very simple. It is. In many ways, the randomization visit is almost identical to the screening visit, other than there's usually not a need for consenting again, unless, and if you watch the screening video, you would know, if between screening and randomization, usually a period of time exists. Sometimes it's as short as 1 day. Sometimes it is the same day. Sometimes it's as long as 45 days or more between screening and randomization. That's called the randomization window. So unless there was a change in the informed consent form from screening to randomization, there's no need for another informed consent at randomization visit. So I'm looking at Creo. We use Creo for my site. It's eSource and eReg. And I'm going through a randomization visit on a study that I have. And, you know, this study is probably indicative of a lot of the studies that we have. It's slightly, almost the same procedures as at screening. There's a few exceptions. One is the informed consent, unless, of course, you have to remember, this is why experience matters in research, because exceptions do pop up here and there, and ignoring these exceptions could be disastrous for your study. So at this study, randomization, we don't need demographics anymore. We don't need informed consent. We don't need medical history, unless there's been a new medical history diagnosis between screening and baseline. And again, anytime you hear me say baseline or randomization, it's the same thing. Also, another common way to refer to baseline and randomization visits are visit 2 or day 1, mainly because visit 2 is the next visit after screening, which is visit 1, or day 1 because it's the first day of the patient actually taking the study drug. So baseline, randomization, however you want to call it, is really important visit. It's probably, arguably, the most important visit that the patient will have from a protocol perspective and a data quality perspective, because it's your last opportunity to really double, triple check, does the patient actually belong in the study? Do they actually meet all the inclusion-exclusion criteria? And it sounds simple. If you've been doing your due diligence between screening and baseline, it should be. But always double check, always have somebody at your site doing quality assurance to make sure that these things are being done. So on this particular study in Creo, we have ConMeds. Have there been new medications since screening? Immediately you want to make sure, okay, is it a prohibited med, or are we still okay with randomizing the patient? And the aforementioned medical history, why were they given this medication? Well, maybe they just took Tylenol 1 day because they had a headache. All right, well, technically that's an adverse event of a headache, and technically it's new medical history, headache, and ConMeds. So some CRAs will tell you, well, don't put headache as medical history, just put it as an adverse event. Other ones will tell you, well, because they took a medication for it, it should go under ConMeds. Every CRO, every sponsor is a little bit different, but just know, very important to know, were there any changes in your medications from screening to baseline that we need to know about? And then how does that affect the protocol, either from a new medical history standpoint or from a prohibited medication safety standpoint or efficacy standpoint. The next thing on my list, this is a pain study. So it's another pain assessment, which is unique to this study, but other specialties, maybe cardiology, will have an angiogram, or hypertension might have systolic reading. That's part of vital signs. Gastroenterology might have another colonoscopy. It could be anything. The protocol-specific assessments, usually whatever you did at screening, are also done at baseline, because they want to compare, screening, they want to compare baseline, and then they want to compare these same efficacy and safety assessments, which are usually the gold standard of that particular therapeutic indication when they design the study. They want to compare those over the visits as the patient is progressing through the study. In this particular study that I have, there are 18 visits, and that is over a period of 8 weeks. So it's like fast-moving, multiple visits a week. In that respect, this is an unusual study, but in the respect of this baseline visit example, it's not. Another assessment we do is physical exam. Physical examination needs to be done for this particular study. Some protocols don't require a physical exam at baseline, but physical exam, it never hurts to do one, even if you don't need to do it. You can argue it's a safety assessment. So if your PI wants to do a physical exam, your PI can do a physical exam. Vital signs is another one we have here. And vital signs is one that's pretty routine across the board. Anytime the patient comes in, they're going to have vital signs. ECG, another one that's pretty standard across the board. Labs. I haven't seen any studies where there's not a lab assessment, meaning blood draws, and safety panels, chemistry, lipid panel, all the lab tests being done again. The same ones that were done at screening are going to be done at baseline, and then at various different time points throughout the study, again, to compare safety and efficacy. And sometimes things like pharmacokinetics and pharmacodynamics. What does the drug do on the body, and what does the body do to the drug? For this study, we also need the actual administration of the study drug. But before the administration of the study drug, that's really the last thing that should be done at a baseline visit. Before the administration of the study drug, because you can't do it until you get into the IRT system, remember, and click the button to say, yes, all the inclusion-exclusion criteria were met. You need to really go and pay attention to the inclusion-exclusion criteria. And let me tell you, those criteria, I'm looking at them right now for this particular study. I have 12 inclusion criteria, and I have, for this particular study, 55 exclusion criteria. And a lot of them are really like in the weeds and getting into like the nitty-gritty details of patients, whether it's labs. In this particular case, it's cortisol levels. There's another one for bilirubin levels. There's a bunch of different specific lab results that they want for this study. There's another one for blood pressure requirements, both at screening and at baseline. So oftentimes, these inclusion-exclusion criteria, it's not enough to just meet them at screening. Patient also has to meet them at baseline prior to randomizing. And yes, you can screen fail a patient at baseline and never randomize them. That's called a screen failure prior to baseline. So you can screen fail from day one of screening, so visit one. I shouldn't say day one because day one is another synonym for baseline. You can screen fail someone at the visit one screening visit. You can screen fail someone anytime during the window between screening and baseline. And you can also screen fail someone at the baseline visit before you actually randomize them, before you actually click the button on the IRT and get them randomized. But this study, look, it's 55 exclusion criteria. Somebody really has to go through one by one on those things. And you really shouldn't do it at baseline visit. You should know the answers to almost all of these things prior to the patient even coming in. But there are some things that you won't know until the patient comes in, namely any efficacy assessments that they have to do in the study, or sometimes safety too. Because remember, I said they get an ECG done at baseline visits. I've had patients screen fail. I've had patients have normal ECGs at screening visits and then have abnormal ones at baseline that were actually part of exclusion criteria. So something as simple as ECG, just because they did well on the ECG at screening visit 1 doesn't mean they're a shoo-in to pass that same assessment at visit 2. Things happen, and clinically significant, not clinically significant. Ultimately, it's up to the principal investigator to decide these things. You can also screen fail for the efficacy assessment. Maybe during the screening window to baseline, there's a series of assessments patients have to do. In pain studies, this is very common. Did their pain improve and without taking any pain meds? Remember, during screening, they're not on any medications. They're usually washed out of prohibited meds, so they shouldn't be responding to whatever their therapeutic indication is. If it's a pain study, they shouldn't be improving in pain too much. If it's a blood pressure study, they shouldn't be improving too much in their blood pressure. If it's an obesity study, they shouldn't have lost a bunch of weight between screening and baseline. Those things usually will be triggers for screen fails at baseline. So really pay attention to those things. And also, like I said, the aforementioned con meds, probably one of the most important things to look at. So make sure that you do your baseline visit in order. Make sure that somebody double checks your work. This is where it's good to have coordinators checking each other's work, especially prior to baseline. And then the part about baseline that's easier than screening is you've probably already logged into all the portals that you're going to need to access for baseline at screening visit. So you shouldn't have issues getting into various portals. The part that's different is the actual IP dispensing and administration. So if it's your first baseline visit for your first patient that you're going to randomize, in a way you're going to feel like it's a brand new screening visit because you've never done it before. You have to know how to get the drug. You have to know how to document it in your log. Remember those master IP logs. You're going to start using those things now. We'll do entire videos on IP accountability. I've done entire videos on IP accountability in the past. So you're going to have to start keeping your logs now. Master investigational product log and individual level subject IP accountability log. There are probably also for your study logs for randomization. And you should really keep this log from screening, the informed consent log, the adverse event log, and then the subject-specific ConMed log and medical history log. So really, once you start screening a patient, you start keeping some logs, and definitely once you randomize a patient, you start keeping other logs, namely investigational product logs. So that's a randomization visit in a nutshell. Again, always double, triple check before you hit yes on that IRT button when it asks you, does the patient meet all inclusion and exclusion criteria? If you are in doubt, call your medical monitor, email your medical monitor. Ideally, you want to do this before the baseline visit where you get waivers perhaps from medical monitors, or when there's like a gray area issue and you're not sure how to handle it. This happens all the time now in studies because they're so complex. The coordinator or the PI or the sub-I, somebody from the staff should email the medical monitor their thoughts on, hey, does this patient still qualify if they're taking this medication for this, or if they washed off of this medication from this date. Usually the medical monitor will be pretty responsive in giving you a response back. Ideally, you don't want to wait till the day of. You want to get these answers prior to, but sometimes it's impossible to, and it becomes an urgent matter. And that's when you as the CRC need to start picking up the phone, emails, calling the CRA, the medical monitor, the clinical trial manager, whomever you can, to let them know, hey, this is urgent. We're about to randomize the patient. We need final guidance on whether we actually can or not. The worst thing you want to do is click yes, patient is randomized, and then realize an hour later, you know what? They shouldn't have been randomized. That's a big deviation, and it will probably require retraining, a CAPA plan. It's probably a safety issue. So just always be careful. It's better to take a deviation and wait an extra day if you're not sure. So just take an out of window deviation. Let's say you have a screening visit window of 45 days, and this is day 45, the last day you can randomize the patient. It's not as bad of a deviation to wait till day 46 to randomize, as long as you're sure that you know you can randomize the patient, as opposed to just randomizing them for the sake of randomizing them and then realizing later you shouldn't have, and they should have been screen failed. So this is the stressful part. Luckily, with studies, you start figuring things out the more people you screen and the more people you randomize. But that's basically it in a nutshell. Don't forget, as soon as you're done with the randomization visit, fill out the appropriate IP logs, subject ID logs, whatever's in your investigator site file. Don't ignore that. Another thing, before you get the drug to the patient, make sure there were no temperature excursions. And if there were, make sure you get approval that you can dispense the drug. So get it from either the medical monitor, the clinical trial manager, the CRA, somebody. Get it in writing. If you get it on the phone, make sure you document it, temperature excursions. And also, after the visit, ideally within 24 hours, but preferably the same day, enter the data in the EDC system. And be ready to answer queries, because as soon as you enter data in those EDC systems, you're going to get queries. Also, make sure the PI signs off on the inclusion-exclusion criteria. Even if there's not a place for the PI to sign it, make them sign it, because they need to show proof that they know what's going on and that they think the patient also qualifies. So make sure you get the PI to at least sign off on the IE criteria or sign off on the visit itself in a timely manner, which is the first C in Alcoa, contemporaneous. So that's it. That's a randomization visit. Thank you so much, Guru Nation. Like, subscribe, comment, share. Remember, Life Boost Coffee. Links below. Bye-bye.